So let's see some outcomes from the Human Genome Project. So obviously Human Genome is the largest genome to be extensively sequenced. It actually provided us a rough draft that actually completed in 2000 and finally in 2003. It costed $3 billion. What we see here is that from the Human Genome Project, important facts that we see here are, first of amongst them is that 95% of the DNA is consisting of non-coding sequences. Human DNA is 98% identical to chimpanzee. Two humans, they just differed by only 0.2%, so they are kind of 99.8% similar. So everybody has 99% similar genome. They have approximately 30,000 genes. Earlier it was an idea that there were around 100,000, but they are not 100,000, so they are around 30,000 genes. So why a big genome but less number of genes? Surprising. So here we see the genome size of different organisms and in comparison to estimated genes. So we see that in human the genome sizes is obviously is 3 billion and we have 30,000 genes. So it's quite sparse. In mouse 2.6, same genes. Weed, which is quite small, Arabidopsis thaliana, 100 million, that has 25,000. So obviously with these organisms we see that the genome size is decreasing, but the number of genes is kind of relatively high. So they have higher gene densities, as we see here in bacterium. So we have 4.6 million and we have 3,200 genes. So kind of human genomes are, if you compare to the other organisms, the genome of those lower organisms are kind of quite compact as compared to humans. Then we see that there is a marked variation in the distribution of different genes, transposable elements, GC content. GC content is important that it gives us an idea about stability of the genome, and people have seen that GC content is also correlated with the presence of coding regions. So it's interesting to study GC content. CPG islands, they are there. Actually some changes happen to these nucleotides in the region, especially where we have the genes, so that increases the proportion of those CPG islands. So if we have CPG islands in some area, we can predict our genes. So later on in our gene prediction we will talk about these things. Obviously there is a variation in recombination rate of different genome segments and genes. So here we look into the pattern of repeats, especially the interspersed repeats, the repeats which are interspersed, not present in the tandem fashion. These are different kind of repeats like lines. We just look into their fraction of the genome they are making. So they are 21% of the human genome, signs make up 13%, retrovirus-like elements are 8%, and transposons are 3%. So total 45% of the human genome is made up of repeats as per the findings of Human Genome Project. Although we see that there is half of the genome is made up of transposable element, but there has been a decline in the overall activity of these elements in the overall hominid lineage. Here we see the distribution of GC nucleotides, for instance. So what they have done here is, we are looking on the x-axis the GC content in terms of the percentages. Towards our y-axis they have actually created different 20 kb size windows. So how many 20 kb size windows are present? There are 2000 to up till 12,000. So it ranges from 0 to 12,000. On the x-axis it ranges from 20% to 70%. So what we have seen here is the distribution of GC contents is kind of skewed towards its right. The mean GC content is around 41, whereas if we look into the mod, so mostly it's these two classes 38 to 39%. So we see in this diagram that there is marked variation in the distribution of GC nucleotides. We also observe that hundreds of the human genes, they are as a result of horizontal transfer from bacteria. Another finding is that segmental duplication is much more frequent in humans than yeast, fly or worm. If we look into a comparative overview, Mutation rate is twice as high in males as compared to the females. More than 1.4 million SNPs, single nucleotide polymorphisms, they have also been identified through Human Genome Project. And genes and or at least their coding regions, they are actually the small fraction of the human DNA. There are small exons which are separated by large introns, interestingly. So mostly the average codons in the exons are around 50 codons, whereas introns, they are exceeding over 10 kbs. This obviously creates a signal to the noise ratio. It's tough to do that for the gene prediction softwares. So obviously those computer programs for direct gene prediction, they have a lot of limitations and issues. So that's why mainly they rely on identification of some similar pattern sequences in the related organisms. So what we have seen here is that human genome is the largest genome to be sequenced, and obviously 99% of the genome of two humans is similar to one another, and coding sequences are actually the small fraction of the total genome